Hi, Bradley.
Hi, how are you?
Hi.
Right here. Susan sent me a question about COMSOL, particular cells in COMSOL. So no, I do not know COMSOL. But yeah, I didn't.
No, nobody knows COMSOL. Run into the conclusion. Yeah. Anyways, I'm still fighting with it. Currently I have to get look at the force and see how it's applied to my model. So I have to have my inputs before I look at my outputs. So I have to change the inputs too. So yay. Anyway.
Susan, did I send you the stuff on a Japanese fellow who's working on eye spots in butterflies?
Oh, I don't think you did.
I didn't. Okay, his name is Joji, Joji Osaki. Wait, let me check it.
Okay, well, sure.
I'll check his name. Let's see.
Anyway, I use ChatGPT for my help desk for COMSOL. It gives me general routines to try to find the exact command.
Okay, I got it. Don't talk. I'll put it in the chat.
Yeah.
You said Joji. That was pretty funny. There's a famous musician. There's a famous singer named Joji. Yeah.
Well, I think it's Joji. Joji.
Jojo. Joji.
I misspelled it.
Oh, okay.
Okay, Joji Otaki. At any rate, he's finding that the eye spots are very similar to what used to be called losing the word right now. Inducers, and that raised, and they seem to be the site of mechanical waves.
Oh, very interesting.
Yeah, so while the butterfly is forming, it's not the wing. This is in the wings. The wings are not flat. They actually have raised points in them.
Oh, well, if you have more information, I'd like to see that.
Okay, he's got a lot of papers already published on it. And he's put some poor student on trying to get Wayne Borland's program running.
Hey Dick, how well is butterfly aviation understood?
How about what?
How well is butterfly aviation understood?
Aviation? I have no idea. There's
how butterflies fly.
Yeah.
How well do we understand that?
I don't know. I saw a very good video on how bumblebees fly, but
the bees are getting all the attention.
Yeah, they're getting all the attention.
Do we not call it aviation? Butterfly flights. Yeah.
It didn't mention butterflies. So I don't know.
There's a lot of attention to other insects, other flying insects like bees, like bees, particularly like honeybees. Maybe like flies, definitely. Butterflies, I'm not so sure. I've never lived with butterflies.
Oh yeah, I know a guy who used to hold a fly and then show it a display.
Yep. I don't know ways to do that.
He'd take electric signals off its brain.
Yeah. That's what Robert de Ruyter and William Bialik down at IU Bloomington, where I did my undergrad, they used to do that. They put the flies.
Yeah, well, this guy died young, I think, in a canoe accident.
Oh goodness. Okay.
We're talking about the 60s here.
I know that they have a lot of work on moths and like how they respond to different stimuli and how they fly, so they can track plumes, and then they watch the moths on camera and track the kinematics. So that's as close as I've ever heard of the
They also have maneuvers to try to avoid things like bats.
Yeah.
Because they fly at night and so do the bats.
Yeah. So it makes it really hard to study. Yeah.
During the summer, we tell the bats are up because we have outdoor lights that are triggered by motion.
Yeah. Yeah. All right. Well, all right. Yeah. So let's welcome to the meeting. It's like we have Hussain and Dick and Susan and Mehul and Pakhi and Morgan. Hello. So yeah, this is the first week of Google Summer of Code, the coding period. So we've had the community period for the last couple weeks, and now we're getting into the coding period, and that starts today. So let me go over the schedule for. Oops. Let me go over the schedule for Summer of Code and see where we are with that. So we had our community period, and today, May 27th, coding officially begins. And so, we're going to have, I think, 2 evaluations over the summer. We're going to have July 8th and going to submit an evaluation for both Pakhi and Mehul. And that's July 12th is the first one. And then the second one is, I think, maybe only one during the work period. But there's also the sort of the end of the program, August 26th through September 2nd. And I'll submit an evaluation then too. So the way this works is that we go through from May 27th to August 26th to September 2nd. Actually, I think the deadline is August 26th for student projects. So what we'll do is we'll work through the summer from May 27th to August 26th. And then that's the deadline for submitting projects. And so as students, you have to submit your own projects, package your code, and then submit a link to Google, and they have to run the program and test it to see if it runs. And then you pass. But then I also have to evaluate you a second time to ensure that you did sufficient work. And so that whole process will end on September 2nd. Now you can actually extend your timeline if you need to. If something comes up during the summer, you'll maybe need to extend it a little bit. So you can extend it anywhere from September 3rd to November 4th. So in the years past, we've had people extend their projects out to the maximum, which is November 4th. And it's possible to do. I don't know if I'd plan for it, but you can basically have an extension. I just have to let the people at INCF know that we're going to extend it. But otherwise, I'll be evaluating you twice. You'll be evaluated in July and then in August. And basically, those are very easy to pass. I just tell them, that you've been doing good work. Of course, hopefully you've been doing good work the whole time. I expect you to. And then, I'll just fill out the form and submit it. And, that's good. So, yeah, that's how the program will work. Hussain says, I can attest Bradley is great with feedback and responsiveness. Yeah. Hussain was a GSoC student in 2022. So yeah, he knows into the process and how it works and everything that you need to do. So, there are a couple things that you should keep in mind before we start the coding period. So the first one is that I have set up a repository, and that is in the devolearn site. So I'm going to put in here. I'm going to put in a link, and this is going to be our repository. So this is our repository here, devograph. Okay, yeah. So this is our repository here, devograph. at Friday's open source meeting, I talked about this a little bit. But basically, both Mehul and Pakhi will be submitting to this repository. So we have a number of directories in this repository, stage 1, stage 2, and stage 3. And those refer to the different stages of the project. So stage 1 is like cell segmentation, and we have some materials from contributors past here, but this is basically, we have things related to the cell tracking challenge, which is a data set you can use. There's some things related to mask segmentation, and then there's some stuff here as well where, you know, if you want to use one of the devolearn toolboxes for segmentation, you can, and all those things are kind of in this. The discussion for that, some of the files for that are in this area. Stage 2 is building graph embeddings, and so there hasn't been as much work done on that. We have a couple of Python notebooks here where you can download them and walk through them. You can do whatever you want to do there. Expect that this is going to be improved over the summer, but we'll see how that goes. And then stage 3 is sort of the back end of what we want to do with the graph embeddings. So Himanshu last summer worked on topological data analysis, and so this is a lot of the work that he did, where he has some notebooks here. He's set up an environment, and he has, this is the README here. README doesn't have a lot, but it's basically tells you how to run the environment file and that sort of thing. So we have a lot of gaps that need to be filled, and of course you have your projects. So I wouldn't necessarily put them in stage 1, stage 2, and stage 3 unless they kind of fit into that. You can create your own directory. So what I would do is fork this repository. So we already have 9 forks. You fork it, and then you go through and you kind of create your own space for what you're doing. So if you're doing hypergraphs, you would say hypergraphs, or you might put some of your work in stage 2 or stage 3. I see that Pakhi already issued a pull request, so I accepted that earlier this morning, so thank you for that contribution. If you're working on something else, you might create a directory for that. So, we'll try to keep this, coordinated. We'll try to make sure, maybe we'll even try to improve the repository this summer, because it is kind of a hard thing to kind of get to or get your head around, because we've had a lot of contributors, and it's just kind of hard to keep everything organized when you have contributors coming into the repository, making pull requests, working on different stages of a project. And this is something that we'll talk about in the open source meetings as well, because we have a lot of, between myself and Jesse, and of course Morgan even, we have a lot of experience with these sorts of things, thinking about them. So that's where we are with Google Summer of Code. And so welcome to the coding period. And I kind of expect you to make weekly pushes to the repository. Basically, there's no rule about that, but basically I want you to keep everything up to date. I don't want things on your local computer, and I want to be able to track your progress. You'll give a progress report either in this meeting or in our Friday meeting. It's up to you. And then we can I can maintain or I can monitor your progress. I can help you maintain your sort of momentum. And if you have problems, of course, we'll be able to discuss those in the Slack during the week. So you don't have to wait until Monday to discuss something. You can do it, as soon as the problem happens, and then we can talk about it, debug things. If you have to, change your schedule, if you have to modify your schedule, then that's something we can talk about as well, because that happens a lot during the coding period. Sometimes people need to rescope their projects. Sometimes they need to change direction, and that's perfectly okay. The whole point of GSoC is to teach you how to run through a project from the beginning to some stopping point and then submit some sort of work product. And then, of course, you'll do things along the way that make, all of that happen. You'll learn all the steps. So one of those steps, of course, is generating code. But your code is, maybe secondary to what you're actually trying to do in your workflow and your schedule. So, try to do a certain amount of stuff each week, generate some code, or, implement something, make pushes to the repository weekly. And then, of course, we want to be able to document things. So I would develop some sort of workflow for documentation, whether that be, having notes that are organized, or, committing something in the repository that's like a README file, and just kind of putting things in, and then, keep, revising and updating it. And then at the end of the summer, you might do a final revision and have that there for people who might come to the repository next year and want to know what you're doing. Because that's something that of course, it's aspirational every year. And every year people fall short because it's not the main purpose of GSoC. And that happens in a lot of projects. It's not just people. There's no magic bullet to making perfect documentation. You might find some readings online that talk about it, but no one's figured that out. But that's an important step in this whole process, is to keep notes of what you're doing. Make sure that you have like a sort of a chain of action. So if I, do some things over the course of a week, you want to document that, but you also want to make it so that people can reproduce what you did. So at the end of the summer, you'll have this software that, you'll want people to be able to run or be able to figure out what's in your repository. And you'll want to make notes as to what those things are, how they work, how they fit together. And that's, like we saw with Himanshu's documentation, he gives you some instructions on how to run the software. And that's one thing you can do. You can also create a Jupyter notebook or a Colab notebook where you have your kernels and you have the code, but you also have, annotations in the code that say what it does. And that's why notebooks exist, because we can put the code in these kernels, but we can run the pieces separately and then say what the pieces do. So you don't just run code in a compiler and not see what's going on. You can interact with it. You can just say, okay, there are 10 lines of code. This does this thing. It outputs this graph. I'll run it, and then it'll show the output. And so that's a nice way to walk people through some of the things you're doing. But it doesn't, it's not the only thing we want to do. We want to have an executable at the end of the summer, ideally. Although the rules on Summer of Code are such that you don't have to do it that way. But in this case, that might be a good sort of stopping point. So, there are a lot of things we can do with our software. We can do things with, changing our schedule, things like that. So just keep up to date with the things that you want to do. If you want to change direction, let me know immediately. If there are things you've kind of discovered that you want to explore more, you can do so, but keep in mind you're on a schedule, and the schedule is pretty tight. And the whole point, again, of the project or of the program is to get you from starting a project to some sort of stopping point in a certain amount of time, and only extend your project if you can't make that end point or that stopping point. Because you could do your project forever if you really wanted to. A lot of projects just continue on, and that's fine, except that they're trying to teach you how to make, find a good stopping point and submit a work product. Because if when you go into wherever you're going, industry, academia, that's something that you're going to do all the time. You're going to have a finite length project, and the point isn't to discover all these new wonderful things. The point is to just find out a couple of things or produce a demo or something and report on it. So that's what the goal of the project is. So any questions about that? Okay, yeah. So some Mehul said thanks. Yeah, again, we'll go through weekly. We'll get I'll give you some time to kind of walk through what you've done over the week, and then we'll ask questions about it, and it'll be a pretty good summer, hopefully. Yeah. Okay. So, did anyone have any updates on their own projects that you wanted to talk about?
Hey, so I'm not participating in GSoC. I'm not a GSoC intern, so I've already spoken with several people here already a few times on the breast cancer CT. The breast cancer computed tomography approach I'm working under with Dick, under Dr. So and Dick and I meet once a week already, so we have our one-on-one meetings. But ultimately, so this is my project. Okay, excuse me. This is my project in which we're taking these advancements in X-ray technology and using it to effectively manage and manipulate how we can steer them in a more dynamic manner. Excuse me. How we can steer them in a more dynamic manner by studying by enhancing the precision of X-ray computed tomography using Janus spheres, at least on the one sphere. But overall, we're more so concerned with more efficient and effective AI and ML approaches to controlling and steering X-ray beams for more dynamic adjustments. And ultimately, we want to use these particles called Janus spheres that can give us a lot more control over the direction of X-ray photons and give us much higher resolution images to reduce the dosage of, say, radiation to which breast cancer patients might be exposed to. And see if we can potentially overcome some challenges, such as say like how devices like at microscopic scales, like how that can be managed in the context of like the scales of devices themselves, or it is really interesting because Dick and I do meet together and discuss the papers and the research. What's being published even nowadays pretty frequently, like in nanotechnology, material science, chemical physics, pushing the boundaries of what's possible to basically refine the an X-ray computed tomography approach, which scientists have more or less been dancing around the question for a century now, and we're getting to this today in 2024. Yeah. So there's my update.
All right, that's good. Thank you. Yeah, you've talked about this a couple times, and so a Janus sphere, for people who don't know what that is, could you describe it? Or
yeah, named after I just thought it was cool. Like the Janus particle is sort of a special type of nanoparticle, or like either a nanoparticle or a microparticle. So they can vary in size from the nanoscale or to the microscale, which is quite a bit bigger. In general, we call something a Janus particle when they have two. I believe it's like two different distinct physical properties. So you can think of like one half of it being hydrophilic and the other half being hydrophobic. Similar to like what maybe you guys have heard of like chemistry classes when it's like oil and water that don't quite mix together. They're non-soluble. I believe that's what it was. It's been a long time since I've studied chemistry. Yeah. So named after the Roman god Janus, who was two-faced, and they would say he'll be able to look to the future and to the present, and that's where the month of January comes from too. Like January being like the shift from one year to the other. So that's kind of interesting. Look up the Roman god Janus, and he is two-faced, the god of the new year, seeing the past and the future, seeing beginnings and ends, seeing the present and I guess what's no longer present either. So whatever sort of dichotomy you can really take advantage of, it's really it really roots back to those principles from ancient Greek and Roman mythos, and that's really fascinating and in a cultural perspective too. So yeah. Anyways, Janus particles. That's what they are. Like nanoparticles, microparticles with two distinct spheres, and we are taking advantage of 3D software too, like Blender, which we did Blender, but I'm moving over to Unreal because Unreal just offers a lot more functionality. So Blender's pretty cool. You can easily just add spheres with a click of a button. You just click a button, you add a sphere to Blender. It's in Blender's the animation software. Yeah.
Yeah, that's great. So yeah, look, be interesting both from like a sort of a imaging perspective, but also from like what you can do in Blender and Unreal in that 3D space and modeling this stuff.
Yeah, I was wondering if Blender might work better than COMSOL.
Oh hell yeah, of course. But so Blender's really, you can do physics simulations in Blender. That is true. You can actually use scripting in Python and C++ to just even simulate things as they are, trajectories of beams for X-ray technology. But Unreal offers a lot more functionality. Unreal's another like 3D engine for graphics processing for game development and things like that. So yeah.
Does it give you enough information so that you could build a bridge so it doesn't fall down? Like, is it a software that's mechanically sound?
Mechanically, okay. So physics, from a physics point of view, yes. For the most part, yeah. I mean, it's okay. Look, it's interesting. It has its own set of physics. It's not the perfect physics of what we talk about in the real world. It's all simulated. Like you can actually recreate like the double slit experiment for one thing, and it doesn't give you the results you should look for. That's kind of funny. But like mechanically, though, okay, you give it the physics that you want to see. That's the thing, right? And it works based off of an engine that you yourself can function and program yourself. Like I can add my own chemical properties to, like if I want to create a bridge, like as you described, like a bridge per se, yeah, and test how it's going to, like how would these materials respond in a different condition, a weather condition that's very, very adverse, You can actually simulate that. You can simulate that. And for the most part, I believe it's pretty sound. Blender is too. I know Blender is used, I think it's been used in forensics, but don't quote me on that, like for certain, like...
Yeah, I've heard of that.
Yeah. Unreal, see, the thing is, Unreal has a lot more functionality, but with that comes the caveats of how true to like reality, how true to the norms of like practical application you're really willing to stay upon. So like that's why, for example, with this project with breast cancer computer tomography approaches, we need to understand, say, the capabilities of medical devices, like that are involved in the X-ray technologies themselves. To what extent can they be used in simulating those things too? So it's not going to do any good to the hospitals and manufacturing plants if I have a solution that's not feasible. So, yeah, of course.
Have you heard of rhinoceros too? Rhinoceros and grasshopper? No.
Wait, you did not just say rhinos?
Yes.
Mm-hmm. Yeah, the rhinoceros.
Yeah, there's a rhinoceros software. I have it actually.
Oh, it's a software. Oh, wow.
Yeah.
First time ever. No. Okay. No.
You can detect segregates using grasshopper, apparently. But it's like Symlink to me from MATLAB. So I don't know.
Rhinoceros 3D. Okay.
I think it's expensive.
Okay. No, I haven't. Yeah, within science, believe it or not, within scientific research, Maya's really hot. Maya's pretty valid too.
Maya's been around a long time.
Like Maya is
yeah.
Yeah, and it's actually Maya does cost money as well too. So you need a student license for that kind of cost. Yeah. Anyways, yeah.
Oh well.
Yeah.
There's a lot of different software out there, and I'm using one that seems to have no help desk, so I keep wondering if I could do something else. Yeah. Well, as to my research, I found someone else simulating epithelial cells. So let's see if I can Yeah, I think there it is. And they've kind of done a weave of the outline of the cell, and it's produced some good results. So now I get to relate my model to this model.
Yeah.
Yeah.
Cool.
I don't know what software they were using. They, papers, they just don't say what software they're using.
Right.
Yeah. I think it's I mean, regardless, like whether you want to simulate it with Blender or Maya, it's good to be, good across different software. Like the software is the software, right? What matters is like, is what you're producing sound? Is it valid? Is it reproducible? Within the constraints of these like simulations, like Blender having its own capacity for Python scripts. That's all you really need. You can give it the Python scripts itself and ensure that whatever happens in your simulation is valid in sound. Like you don't have to be so certain that is this software like Blender, Maya, Unreal? Is it like going to translate into real results? Well, if you can script it in Python, you can show that there. Then that's what.
Yeah, that's what matters.
So one thing, although like excuse me, I think well, I think translating the results though from simulations from the software into real world practical applications, there's still this like I don't know, it's more so like because like let's say like whether I'm doing it with Blender and it's like, oh, Blender's not really working out so much, can I switch over to Unreal? Like the more you can take advantage of, like the more you yourself can control within those environments, that's also what helps too. So like
Yeah. Well, COMSOL comes complete with the physics.
Okay.
Like it's finite element analysis physics. So, yeah, and tensegrity doesn't quite fit with it. It's not exact. It's just, anyway.
Yeah.
Yeah, that's great. Yeah, looks interesting. The multi-scale paper here. So static and dynamic mechanics of cell monolayers, a multi-scale structural model. So looks interesting.
Yeah, it has some good quotes for me in it, and I'm only going to do half of what they were trying to do with their model. So we'll see. I'll do that half, and then maybe I'll get told, no, do the rest. Maybe get ordered around, and they'll say, no, do all that. Yeah. I can hear it happening already, but I got to do the first part first, like the static analysis first.
Yeah.
Oh well, so that's my project. I have to look at inputs this week.
Okay.
Find out actually what's going on with the inputs.
Do we have any other updates from anyone?
Speaking of which, like, there's also been this shift towards Unreal as well too, towards Unreal and Maya. Part of a lot of companies shifting away from Unity. If you were there about a year and a half ago, there was this big debacle with the CEO of Unity. Yeah, and the company so like that was difficult because Unity used to be something we'd all learned for development and processing, and so there's shifts away from it. But there was like this. They made some very poor business decisions. I'll just say that. So.
Yeah. So, okay, so I'm gonna go to a paper, and this is something that Dick sent. So this one is actually on C. elegans, and so what they're trying to do in this paper is they're using this technique to look at cell-specific proteomics and interactomics, and they'll talk about what that is here. And they're using a specific marker to sort of get at or classify different types of cells. We'll see what they're doing here. So let me zoom in here. Okay. So this paper is MACSPI or MACSPI, enables tissue-selective proteomic and interactomic analyses in multicellular organisms. So they're using that. They're using this method to discover tissue-selective markers, and they're looking at this in C. elegans. So let's take a look at this. The abstract reads, multicellular organisms are composed of many tissue types that have distinct morphologies and functions, which are largely driven by specialized proteomes and interactomes. So, one of the things we've been interested in this group is differentiation and building differentiation trees. And so, the question is how do we make those determinations using what markers? You can look at the phenotype of a cell and you can tell whether something is differentiated. So for example, you can look at a stem cell, you can look at a neuron or maybe some epithelial cell, and you see differences between them. And we have, we can characterize the shape of the cell in, say, a microscopy image, and we can determine when that differentiation happened and under what conditions. You can also look at the molecular markers, which are harder because, we don't have a good recipe for, every change that goes into making, say, an epithelial cell. We know the markers, the candidate markers, but it's hard to know under which conditions that transition actually happens, what are the precursors, and so forth. So in the literature, they have candidate genes, candidate proteins, but beyond that, we don't really know what to look at and what the thresholds of expression are to know what defines a differentiation event. So, we're working on a paper along those lines, and it's pretty hard to determine. But what they're doing here is they're using a new technique. So they're developing this localized proteomics approach called methionine analog-based cell-specific proteomics and interactomics, or MACSPI. And so that's what the acronym is in reference to. This method uses the tissue-specific expression of an engineered methional tRNA synthetase to label proteins. And so the methional tRNA synthetase is a biomolecule. It's engineered for this specific purpose. The purpose is to label proteins with a bifunctional amino acid in selected cells. So you're basically engineering a protein that will express in specific types of cells. It's easy to recognize when that's being expressed, and so it's easy to use as a marker. And so whenever it's expressed, we say that's our marker. That's where we determine whether this has been expressed or not. And then we can, look at a microscopy image, see where the marker is expressed. We can get spatial information, temporal information, and all of that. We applied MACSPI in C. elegans, a model multicellular organism, to selectively label, capture, and profile the proteomes of the body wall muscle and the nervous system. So if you're not familiar with the C. elegans morphology in the adult or even in the larval worms, you have body wall muscle, which is along the walls of the body. It's usually the muscle tissue there, not the epithelial tissue. And then the nervous system, which is, of course, all of the neurons, both in the central nervous system. There's some neurons in the tail and some nervous system cells along the lateral line of the worm. So this is what they're profiling. So they're not profiling every cell. They're just looking at body wall muscle in the nervous system. So they're looking at the proteomes of these cells. And we know all of the cells in the morphology of C. elegans. That's what open-worm is all about. And so they're just taking a small subset of those cells and looking at the proteomic profile. Now, in doing so, it leads them to the identification of tissue-specific proteins. So you can use these markers to sort of select cells. You can also identify tissue-specific proteins because these are specific types of cells. These are muscle cells, neurons, And muscle cells and neurons are closely linked in terms of their composition. So they're not the same cell, but they they have like similar markers. They're both kind of excitable cell types. So this is restricting it to that as opposed to looking at germ cells or epithelial cells can help because you're not your profile is restricted. So this is what they're doing with this method, using the photo crosslinker, which is a way that they able to identify proteins and do things like that. We successfully profiled HSP90 interactors in muscles and neurons, and I'll talk about HSP90 in a minute, and identify tissue-specific interactors and stress-related interactors. So what they're trying to do is they're trying to get a sense of what proteins are interacting under certain conditions. And of course, if you go into the literature on molecular biology, we're all familiar with genes and gene expression, and that genes make proteins, and that multiple genes make single proteins, and that proteins can be modified, transcripts can be modified. So there's all this complexity. But then you have these interactions. So once proteins are formed, they interact. They, and you have to kind of disentangle those interactions to understand what the proteins do and what their abundances mean in terms of function. So this is something that happens in certain contexts of the organism. So sometimes we have very specific interactions related to tissue type. So in muscle cells or neurons, or even in epithelial cells, we have specific interacting proteins. We have specific interactions that occur. And then we have things that are related to function. And so there's, the stress response is very common in organisms and involves a lot of proteins. And so but that's something that's somewhat well characterized. So we can look at stress-related interactors as well. And if there's a stress response in the organism, it could be due, we think about stress, we think about if you're under stress, psychological stress, mechanical stress. But in biology, there's a lot of stress-related stuff that happens whenever the organism is maybe undergoing a change or it's challenged by its environment. So if it's in there's something called heat shock, which is where the organism is exposed to a high level of heat, and especially in development, that can trigger a lot of these stress pathways. And so that's an important thing to look at because it facilitates a lot of changes that otherwise you wouldn't be able to observe. Our study demonstrates that MACSPI can be used to profile tissue-specific proteomes and interactomes in intact multicellular organisms, and so they're using C. elegans as a way to sort of characterize this. We know the cells, we know what they do, and we know the sort of the stress-related response. Now, there's this thing called HSP90, and this is the Wikipedia page for HSP90. So as I mentioned before, we're interested in the stress response. It's sort of a perturbation of the tissue or of the developmental system. And this is a well-characterized, what they call chaperone protein. And so heat shock protein 90, as we said, heat shock is a form of perturbation, a form of this sort of stress response. If you expose an embryo to heat or an organism to heat over a long period of time, it'll challenge the organism's physiology, and their physiology will respond in kind. So in yeast, in C. elegans, in Drosophila, you'll see this heat shock response.
You get heat shock proteins expressed due to alcohol and microgravity. So other things cause it to be expressed.
Oh, yeah. And there are other heat shock proteins besides HSP90. This is just the one that they focus on with these heat shock experiments. So yeah, stress is you can have you can expose an organism to all sorts of things, and you can exhibit a similar response. So it exhibits a similar response. So yeah, there are these chaperone proteins that are involved in sort of assisting other proteins to fold or unfold. So this is an example of these kind of interactions that are occurring in the proteome. There's the chaperone protein that moves with another protein to help it fold. And if you express it in different amounts, it can facilitate or hinder that response or that set of interactions. So if you have a heat shock, HSP90, is it assisting proteins to fold or not fold? If it's there or not there, it can't do its job, or it can do its job more effectively. So they help proteins fold properly, stabilize proteins against heat stress. So when HSP90 is there, proteins can fold properly even in the face of this heat shock. And then also it helps clear old proteins that need to be degraded. If you know about one of the hypotheses about neurodegeneration, they're sometimes, you have problems with degrading old proteins. And so this is a problem that HSP90 can help with as well. So this is why, people have explored the system quite a bit. And so this is basically talks about heat shock proteins. So there's a class of these kind of proteins. They're the most highly expressed cellular proteins across all species. And they account for 1 to 2% of total protein in unstressed cells. However, when cells are heated, the fraction of heat shock proteins increases to 4 to 6% of cellular proteins. So you can see if you induce this heat shock that you get a lot more of these type of proteins. And, this is interesting because, when we talk about differentiation, is differentiation in a sense maybe, or is it maybe induced by a stress-like event or a heat shock event or something like that? So in other words, when you have this kind of response, does it enhance differentiation, or does it trigger differentiation, or or is differentiation just simply something that isn't involving these kinds of changes in something like a heat shock protein? So I mean, it's an interesting thing. I know that people have done experiments where they've looked at differentiation being induced under certain conditions. So we know, for example, in cancer cells, that they differentiate down a cancer pathway in stressful environments, that stressful environments can trigger those events. So that's an interesting point to kind of think about, this relationship between these kinds of protein-protein interactions and differentiation. Whether the differentiation is sort of normal, like you would see in development, or whether it's abnormal, like a cancer, going down a cancer pathway, or an apoptotic pathway where the cells die prematurely, and I guess it would be a necrotic pathway. But they've done a lot of this experiments with this type of thing, but I wanted to mention that as context for this paper. So what is the significance of this? So multicellular organisms synthesize a specific set of proteins in each cell type to enable cellular differentiation and functions. These proteins interact with each other to form complexes, and this is needed for various cellular processes. So as we differentiate from one cell type to another, our cell processes change, and the complement of proteins changes with that. So if we can monitor the proteome, and especially these protein-protein interactions, we can get a better handle on what proteins are involved in specific cell type differentiation. And so there are very few methods that can really robustly profile cell-specific proteomes. We can profile proteomes and protein-protein interactions in bulk, but getting these cell-specific proteomes and understanding the sort of specific markers involved in, say, like muscle or neuron or whatever, those are a little harder to get at. And you have to really kind of to do that, you really have to physically isolate the cells, do like single cell analysis. And that can be a problem biologically because it's not in the biological context. It's just kind of like, you have a muscle cell. After it's differentiated, you look at the complement of proteins, but proteins are very unstable in that way. They don't maintain their profile. If you're in context of an organism, for example, you have proteins that are responding to not only the cell type, but the physiology and everything. And when you take the cell out of that context, it can change things quite a bit. That's also true of gene expression, by the way, but that's a different topic for a different time. So they've developed this chemical biology approach to label proteins from specific cells. This carries both a biorthogonal tag for affinity purification and a photo crosslinker for capturing these interactions. So this is so they apply this to C. elegans, and they say that profiling tissue-specific proteomes and interactomes is particularly challenging in small-sized multicellular organisms such as C. elegans due to the difficulties of isolating large numbers of cells and maintaining the integrity of cellular structures during the physical isolation. So this is where C. elegans has just under 1,000 cells in the hermaphrodite, and cell specific or function specific cells, or distinct types of cells, you have even fewer. So being able to profile cells by their cell type is hard in C. elegans as opposed to something like in humans or in mammals where you have maybe like in the liver where you have a lot of cells of a certain type, and then they're being regenerated and so forth. So this is definitely harder to do in C. elegans, but we also know exactly what the cells are. So they've developed this method. They're able to give some results here. They kind of show what they're doing here. So this is where cells are expressing this wild type MET-RS, and they show that it can be whether it can be labeled or not. And this is the phenotype of the worm. So they're doing this in certain cells, and they're showing sort of how this labeling process works. That you have this cell-specific proteomics, you have these cell-specific protein-protein interactions in the cell of interest. And they show a lot of, like molecular biology here, a lot of stains. And they show kind of, they do these validation steps where they stain things and they kind of look at different things being expressed and different markers. So you want to be able to get the right marker. You want to be able to target the right cells, target the right signal. So that's what they're trying to do here. And they actually show some of the things that they're doing in terms of engineering these markers. So, they're doing some protein sequence analysis, and that's what that is. And so then they show kind of like this result here where they're able to identify sort of the content of muscle and neurons, so they're able to differentiate between the two cell types. And of course, there's overlap between the two cell types. And you have different genes being expressed in neurons and muscle. So you have these, they're identifying this by gene along for these two cells. So let's see if I can find the in the figure caption here. So C is a Venn diagram showing the overlap of identified HSP90 interactors from body wall muscle and neurons. So this is where in C, where we have muscle and neurons, where we have unique interactors, and then interactors that sort of overlap between the two cell types. Not surprisingly, since they're, have some similar functions and might have a similar profile. And then D, two-dimensional scatter plot of proteins identified and quantified using this HL ratio, and the HSP90 interactomes for different tissues. So these are actually, these are proteins, not genes. And so they identify these specific protein types in muscle and neuron, and they plotted according to this metric HL. And so you can see that there's some outliers here that they identify by name. And then there's this cluster here around 0. So yeah, there it's not, they've been able to identify specific markers that are different proteins that are outside of this cluster. And so those are things of interest. And so E here is GoTerm enrichment analysis. So that's where they take like a protein and they give it an annotation. So they give it like sort of its functional. They basically have done these. They have these GoTerms where they have the name of the protein or the name of the gene, and they have like a function attached to it. And they're usually not very specific, but they do give you some information about what that protein or gene does. So they're using these GoTerms of identified proteins from body wall muscle and neurons. Only significant p-values, significant GoTerms are shown. And so this is sort of a profile of these, like basically these functions. So they have these proteins and they have some related function, and they're in different parts of the, different functional space. So for example, you have a bunch of GoTerms related to biological process, a bunch of GoTerms related to cellular component, and a bunch of GoTerms related to molecular function. And so here in this part, we see that there's this p-value minus log 10, which shows the difference between muscles and neurons. So you see these types of things expressed in neurons and expressed in muscles. So for example, glycotic processes are expressed in muscle, IR1 mediated unfolding protein response expressed in muscle, lipid transport is specific to neurons, and so forth. Translation, of course, is specific to both, as you might expect. Ribosome is specific to both, and so on and so forth. So you can get a sense of sort of the functions that are cell specific based on their expression and how we can characterize these. But to get all this information, we need this sort of this marker, this probe that we can apply to the cells, and we can actually get this information. Okay, so that's all. I think that was a good paper to kind of go over to talk about sort of the complexities of, looking at something like proteomics and cell differentiation.
10 o'clock.
Need to look more at that. That's great though.
Yeah. And again, this is just like one way to do this, and the innovation here is to have like a probe, a marker that allows us to sort of make these, sort of get at these interactions and where they're happening and what kinds of things are involved in them. And so it really kind of pares down our search space for the things that are relevant to cell type. And so, again, it doesn't tell us about the differentiation process per se, but it does tell us the things that are implicated in cell types. What are they? What is going on in terms of, when you have different cell types, obviously we know that they have different functions. A muscle has a function of moving the organism, basically. Neurons have the function of sending a signal to the muscle, sending a signal to other neurons. But then what are the proteins that are involved and what are their functions? And so that's where we're getting an innovation
here. Excuse me. Yeah, absolutely. Just how hard it is to, track all that complexity.
Yeah.
Again, like kind of just reiterating things about the. How much you can get from structural imaging about cell function and physiology, and how important it is to track all this complex protein interaction and expression, and how dynamic it is.
Yeah, definitely. And this is just a snapshot. Yeah.
Yeah. I mean, this is just a snapshot. I posted a paper for a protocol from Alison Luttree in Brain Organoids.
Okay.
And it's talking about, so one, it's just nice to see a protocol from him. And two, he calls it a semi-guided protocol. And what he's saying is it's a protocol to generate his kind of electrophysiological oscillations organoid. And is talking about the difference between the unguided and the guided, like having some guidance, which is just some of the chemical baths that you're using, right? And how just how that's changing, giving that extra those extra differentiation signals. And it would be great to see like how to capture how those cells are different now, in terms of this semi-guided and hence they're having much more structured electrophysiology.
Yeah.
It's these this kind of proteomics is what we need to match with this kind of organoid, these organoid platforms.
Yeah, definitely. Especially like in organoids, I mean, you're doing a lot of directed differentiation. Like it's very unlike maybe like biology, especially if you're introducing stresses. Right. And yeah.
I mean, I still like the kind of the Levin argument of it's all biology.
Yeah.
But I know what you mean. And yeah, it's what we know how to control or what we know how to direct. But but certainly, like I still need help with what are the tools you can use to capture the even in snapshots to capture the differences in, say, the unguided and guided. And things that can do more, that can capture more, well, a wider range of activity or expression.
Right. Yeah, that's the thing, is like getting the realistic activity or that wide range of activity. You want methods that allow you to capture that and not have like a very constricted view of the cell. Like, a lot of laboratory methods suffer from that, where you have this, like, I'm going to idealize the conditions, but that's not the way you usually see in biology. So you're going to end up with like, a limited subset of the things that are involved. So
they can come up with some pretty quite frankly bizarre conclusions at times if you're overly constrained. So
yeah.
Appreciate you sharing all this. I would say I've seen a lot of the limitations, particularly with size itself. And I mean like literally like physical size. So yeah.
Okay. Well, if that's all for today, any other things you wanted to say before we go?
Yeah. So I will most likely be moving out to California for my job. So for those of you who don't work, for those of you who don't know, I've been working at a startup, which is we're very, very close to the stages of no longer being called a startup and actually being called a business. I work for this place called Alter Learning. They're based in California, an educational technology startup where we create Steam games for kids to play in VR, and we reach out to K-12 schools to educate them. And this is like it's going to be a curricula, a standard for high schools across the nation, across the United States, for VR games in Steam. We've had this company together for years, and we're just now getting off the grounds now. So, we've been through the ups and downs, we've been through the red tape, signed on things with the curricula. We've been receiving offers from investors, and it's so exciting that I can be part of this, like this startup, and be part of this really, really great, amazing thing for the United States and for the world moving forward too. Yeah, we really have to. This is why I've had to really keep these things like not. I mean, I am under NDA, but like that's why I can't necessarily jump the gun and say I'm starting the next Apple right now. We had to go through everything to make sure everything was put together. And for me, sitting here in my house here in Indiana, I can now move out to. I basically, for me to continue as a full-time employee, I got to move out to Davis, California. So it'll be really exciting. I'll most likely choose to live in Sacramento. So for those of you who aren't familiar, Sacramento and Davis are just about 15. Like it's pretty close.
Yeah.
Isn't it 10 miles? It's something like that.
Very close.
Very, very close. Sacramento and Davis. And I'm pretty sure housing is more affordable in Sacramento. So that's ultimately why. But yeah, they come over based in Davis, California. And I'll be moving over to Sacramento to work full time. And I only have to commute to the office 2 days a week. And that's going to be pretty damn good. So yeah, I'm going to be much happier on the West Coast, speaking of which. The Midwest, I love the Midwest. I love Indiana and Illinois too, where orthogonal is. But like, I'm more of a California person, more of a West Coast person. So anyways, excuse me. Yeah, that's some pretty exciting stuff. And speaking of like, where things could go with, yeah, thank you, Zach. Where things could go with like what work I could be able to do. So that means I could be able to reach out and network with anybody on the West Coast, and the West Coast as well. So yeah.
San Francisco is still pretty close.
If you want to know something about the Canadian curriculum, I taught high school science and math for 11 years.
Yeah.
So I know the Manitoba curriculum, which is different from the other provinces. So if you're...
And obviously different from America too, So.
Yes, yeah, it's similar. But
things can... You'd be surprised. I mean, maybe you wouldn't be surprised, but like things depend on political stuff, cultural things, the views and the needs of schools. I know what I was taught from a scientific perspective, from science and maths and things. So I went to high school in Indiana, this city here called like Zionsville High School, and what they would teach, like not just scientifically, like in terms of like what you'd study, but how curriculum depends on a lot of stuff that can be more like, I don't know, policy tape, like red tape based, like, sort of thing. So it's really interesting. So to see where things go. But yeah, from a tech, neurotech, biotech perspective, I'll be moving over to the West Coast pretty soon. So yeah.
Yeah.
Anyway, yeah.
Congratulations.
Thank you.
Yeah.
Okay. All right. Thank you. See you next week. Bye.
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 Take care.
Take care.